Hi, thanks again for joining us. And this is Zheng, and I'm a product specialist in the Novogene, and I'm responsible for the Epigenetic products. And so the topic of today's webinar is a beginner's guide for the ChIP-Seq and the RIP-Seq. So I will go through the whole pipeline from the sample submission to the bioinformatic analysis to see how the Novogene can contribute in your scientific research. So firstly, I will give an introduction to the workflow of the ChIP-Seq and the RIP-Seq. And then I will show our standard analysis content for the ChIP-Seq. So the RIP-Seq have very similar content as the ChIP-Seq, but focus on the transcriptions. But I will not share them both since due to the time limitation. And finally, a case study will be shared, and which is a published article that powered by Novogene. So let's move on to the first part, the ChIP-Seq. So firstly, I will give introduction to the gene transcription regulation. So the conversion from the DNA to the RNA is called the gene transcription. And while the genetic code determines the translation results, and the epigenetic can regulate the gene expression level and lead to a phenotype change. So before the transcription start site, as we can see, there is a region called the promoter region. So the protein binding here can regulate the further transcription. And those proteins include the histone modification and transcription factors and other DNA-associated proteins. And the ChIP-Seq can provide us with the genome-wide profiling of the DNA that targets by those proteins. And here is the principle of the ChIP-Seq. So the ChIP-Seq is the Chromatin Immunoprecipitation, and it's a method used to analyze the DNA interactions with the proteins. So here, as we can see, the step one will be the cross-linking. So we will use the formaldehyde to fix the protein to the DNA. And step two will be the chromatin digestion. So either use the sonication or the enzymatic digestion are okay. And after the fragmentation, move to the step three, is the immunoprecipitation. And we will use the specific antibodies that against to the proteins that we are interested in and wash away other protein and DNA. And step four will be the cross-linking reversal. So we separate the protein and the DNA. So we will add the adapter to those DNA fragments and conduct the sequencing. And here is the project workflow. So after the fragmentation, it will be separated to three groups. So the input control group, the IP group, and the IgG group. So in the input control group, there is no antibodies, and they are just the DNA fragments. And for the IP group, we use the specific antibody to against the protein. And for the IgG group, they are negative control antibody. So it cannot specifically binding to the protein, but it's the same type as the IP group use. And both the input control group and the IgG group are aimed to exclude the false positive results. And before handing them to us, and we also need a DNA purification. And all those parts need to be done on our customer's end. And the Novogene will be responsible for the lab preparation and the sequencing. And also we can provide the data analysis if needed. And here is the study design. So we will recommend the 2 biological replicate and for each sample. And as we can see, each condition we will need one sample, one input, one IP sample. And for the input control group is required, and we need one sample for each condition. And if you are interested in more than one protein at the same time, and one input sample for each condition is enough. And here is the IgG control group. they are optional, and 1 to 2 samples for the whole project will be enough. So here is a workflow, and after the sample preparation, we will conduct the sample quality control, and then we'll conduct the library preparations and do internally library quality control, and then do the sequencing. And before we release the data to you, we will do data quality control. And also we can provide the bioinformatic analysis for our clients. And here is the sample requirements for the ChIP-Seq. So we only accept the IP DNA, and we will recommend at least more than 10 nanogram and strongly recommend more than 30 nanogram. So the concentration should be more than 2 nanogram per microliter. And if the bioinformatic analysis needed from the Novogene, we will require a diploid species and should have a reference genome with the gene annotation complete. And here is our example of the sample quality control results. And so after we receive the IP DNA from you, and we will proceed 2 steps. So we will use the qubit to quantify the DNA concentration. And then we will find the DNA fragment size distribution by use the agarose gel electrophoresis. And as we can see, both of them are the quantified results. So the difference is the first one used the sonication digestion. So there are 2 similar brand. And for the enzymatic digestion, due to they have enzymatic digestion site, so the results were like this. And a quantified result, the brightness between the 100 to 500 base pair should significantly higher than the brightness above 500 base pair. And also the agarose gel electrophoresis has the requirements. So the amount should more than 20 nanogram. So if you send us with the sample less than 20 nanogram, you need to decide whether to proceed the library preparation will only based on the qubit results. So after the sample quality control, we will conduct the library construction. So for the IP DNA, we will do the end repairing and add the adapter. So then we will do the PCR amplification. And after the site selection, we will get 200 to 400 base pair in the DNA library at the end. So after the library is ready, we will then do the sequencing by using the pair end 150 in our Novogene 6000 platform. And before I introduce that part, let's move on to the RIP-Seq first. So while the ChIP-Seq was focused on the transcription from the DNA to the RNA, the RIP-Seq will focus on the translation from the RNA to the proteins. And here is the figure shows that an RNA life. So as we can see, the RNA bursts and get mature and will export from the nucleus to the cytoplasm, and then will finally translation to the proteins. And all those steps will be assisted by the RNA binding proteins, which is called the RBPS. And the RIP-Seq was aimed to research on those proteins. And here is the principle of the RIP-Seq. So as we can see, the ChIP-Seq is more like the ChIP-Seq. So they need the cross-linking and the digestion, and they will got an RNA fragment at the end. But the RIP-Seq, they only need the immunoprecipitation and will got the full-length RNA. And so for the RNA fragment, they usually less than 200 base pair in ChIP-Seq. So it has risk on the library preparation. So we will more recommend the RIP-Seq. And here is the project workflow. So it's very similar with the ChIP-Seq. So the only difference is that we recommend an rRNA removal before send the IP RNA to us. And here is the sample requirements for the RIP-Seq. And we only accept IP RNA, and the required amount should more than 100 nanogram. And the 100 nanogram is only enough for one-time lab preparation. And for the RNA integrity number, we will recommend more than 8 for animals and more than 7 for plants and fungi. And also we will recommend the unfragmented sample, and the min peak should more than 1,000 base pair. And for fragment samples, we can accept, and the peak should lower than 500 base pair, and they will be determined as a hold in our sample quantity control results. And the concentration should more than 5 nanogram per microliter. And here are the sample example of the sample quantity control for the IP RNA. So we will mainly proceed 3 steps. The first step will be the nanodrop to quantify the RNA concentration and the purity, and then use the Agilent 2100 to detect the RNA integrity and show the fragment size distribution like this. And finally, we will use the agarose gel electrophoresis test the purity of the samples. So quantified results should like those, and the unfragmented samples, the min peak should more than 1,000 base pair. And for lower than 1,000 base pair, they will be determined as a hold. And here is the library construction and the sequencing. So we will receive the IP RNA, and we will do the fragmentation first, and then use the reverse transcription to get the cDNA. And we will do the repairing and add the adapter. So after the PCR amplification, and we will do the set selection. So we will keep 250 to 300 base pair in the cDNA library. So the information above is all the steps that Novogene will done before the sequencing. So after we got the data by using the NovaSeq 6000, we will then move to the data quality control and the bioinformatic analysis. So here is our analysis pipeline. So after we got the raw data, we will do the data quality control. And we will then do the mapping, and at the same time, we will do the ICC analysis, which is the strand cross correlation analysis. And all those parts are aimed to quantify your data and your IPSA. So let's move on to this first part. And here is our data quality control results. And the E stands for the sequence error rate. And the Q Fred, which is the Fred quantity, is equal to -10 logarithm of E base 10. And as we can see, the Q30 means that the error rate is 1 of 1,000, and the correct rate is 99.9%. And as we can see, the quantity score will slightly drop, and the error rate will slightly increase due to the consumption of the reagents. But we can guarantee that overall, more than 85% data can reach the Q30 level. And after we got the raw rates, we will do the filtration to get the clean rates if the bioinformatic analysis needed. And we will filter out those, include the rates containing the adapters, and the rates containing that more than 10 belongs to an uncertain basis, and also the rates with the low quality. And all the rates sequence will be stored in the FASTQ format. And this pie chart shows that the distribution of all rates. And after the data quality control, we will do the mapping. So we will show the information of all the reads in pair or in read 1 and read 2 separately. And we will give the information that includes the clean reads number, the mapped reads, the unique mapped reads, and the duplicate unique mapped reads. And here is the distribution of the reads mapped to the gene. And since the binding sites of the protein are important for the gene regulation, so the analysis of the relevant, the mapping position distribution can help us predict the protein function. And another important thing during the ChIP-Seq analysis is the ratio of the unique sequence in the total sequence number. So here, the duplicate ratio are shown along with the mapping quantity. So the mapping quantity 16 is the highest. And here we can show the ratio for the non-duplicate, duplicate, and unmapped. And also we have table to show all the reads number. And here is the ICC analysis. So the alignment of the reads to the genome will result in 2 peaks. So one in the sense strand and one in the antisense strand. So it will locate on the both sides of the proteins. And so there is a distance between the 2 peaks. And and as we can see, the second peak is represent the fragment length, and the first peak will represent the read length. And the red line is for the IP group, and the blue line is for the input control group. And here is the quantified results. And if there is no obvious peak can be found here, and it means that your IP assay was failed. So after quantify your data and your IP assay, and then we will move on to the peak calling, the peak notation, and the functional analysis. And those will really help in your scientific research. And here is the peak calling. And as we can see, we need to know that not all the peak can represent the protein binding size, but where the difference can be found between the IP group and the input control group. So they are the really protein binding size, and they are not protein binding size. So that's why the input control group is essential for our ChIP-Seq. And also we will share all the information about all the peaks. So the first one is the peak width, represents the length of the DNA that is bound by the studied proteins. And this one is the distribution of the fold enrichment. So the larger the value, the more reads are enriched in this peak. And here is the significance of the peak. I indicate the credibility of the peak. And this is a summary of the genome-wide distribution of peaks. So as we can see, the vertical axis is for the chromosome, and 23 chromosome, and the green one is actually the protein binding size. And since the protein binding to the DNA is not random, but has the sequence preference, so we will do the motif. So we will use those sequence logo to show the base bias in the different positions in the binding site in the long motif and the short motif. The right side is the reverse complementary to the left side. And we will have 2 different algorithms for the different motif lengths. And as a result, we will share the binding preference of the proteins that you are interested in. And here we move to the peak notation. So this is the peak TSS distance, this transcription start site. So according to the protein binding size, the effect of the iPSA can be detected. And through the characteristic of the protein binding, the regulation mode or the function of the protein can be predicted. And this figure shows the distribution of the peak in the different functional region. And also we will show all the location of the protein binding site. So there's a start site and the end site. And also we will give the downstream genes that may regulate by this promoter region. And then here is the functional analysis. So the horizontal axis is the number of the peak overlapping gene. And the different color is used to differentiate the biological process, the cellular components, and the molecule function. And this can directly reflect the distribution of the number of the peak overlapping gene that are enriched in the functional region. And here is the KEGG analysis. So the vertical axis represents the pathway's name, and the horizontal axis represents the reach factor, and the size of the dot represents the number of the overlapping peak genes and in this pathway. And the color of the dot is corresponding to the different Q value. So comparing the whole genome background, so you can identify the significantly enriched metabolic pathways or the signal transduction pathway associated with the peak overlapping genes. And for more than one group, we can do the comparison. And that's what we can get for you. And so the first one is the differential expression Venn diagram. And it can illustrate the differential peaks of the compared group. And here is the heat map. So those different colors will represent the different clustering information. And the more close between the IP sample and the more similar functions or the biological process they have. And here are the functional analysis, but this time they are for the differential peak related genes. So the analysis content I mentioned above are just the common one. So, and there are also many other diagrams. And due to the limited time, I will not explain all of them. And if you want to know more details, we have demo report, which has the detailed explanations and the notes for all the diagram. And also we accept the custom analysis. As long as you can provide us with the articles, we can evaluate if those analyses can be done by the Novogene. And then I will introduce a case study, and which is an article that powered by Novogene, and to see how the ChIP-Seq and the RIP-Seq can be used in solve the epigenetic problems. This article is published in the Biomedicine and the Pharmacotherapy. So the title of this article is the H3K27 trimethylation loss will play a vital role in the CEMIP-mediated carcinogenesis and the progression of the breast cancer with the poor prognosis. So at the beginning, I will give some background for this article. So the breast cancer is the highest accidence of the malignancy in women. So the breast cancer can be mainly classified into at least 4 clinical relevant subtypes, and based on the presence or the absence of those receptors. And so the triple negative breast cancer, which have no receptors, so due to the lack specific targets, and is prone to the metastasis. And this time we will most focused on the triple-negative breast cancer. And the 5-year survival rate of the triple-negative breast cancer was being found the lowest among all those breast cancer subtypes. So before I share the study results, and I will give an introduction to the study design about why they choose the ChIP-Seq and the mRNA-Seq to achieve his goals. And so from the previous studies, so besides the genetic change, the epigenetic alterations, and such as the histone modifications, have been found to play a role in the breast cancer. So at the beginning, they want to find which histone modification may play a role in the triple-negative breast cancer. So they perform the ChIP-Seq and with the different subtype of the breast cancer cells line. And after they found the histone, and they will find the downstream gene in this histone modification, and to find out which gene may really related to the triple-negative breast cancer. And after they find the target genes, they will need to find a therapeutic drug that may help in the treatment. So they will do the chemical screen. They will treat the various small molecule drugs to the cancer cells, and it's in vitro study. So after that, they will do a vivo study to confirm the drug effect. So use the transfer chamber assay and the migration assay to see the invention and the migration. Also, they will choose the human xenograft and compare the treatment group with the non-treatment group to see the in vivo drug effects. So after they find a therapeutic drug, they need to know that if this drug can actually reduce the target gene's expression. So we found genes here, and we test if the drug has some relationship with the gene expressions. So they will use the mRNA-Seq, and they will also compare the treatment group with the non-treatment group. And finally, to find the molecule mechanism, they will use another ChIP-Seq, but this time they will compare between the treatment group and the non-treatment group. And here are the study results. So the H3K27 trimethylation, and which is a histone modification that can mediate the gene silencing, and the elevated expression of the H3K27 trimethylation was being found to be associated with better survival. And so to verify this, and the different subtype of the breast cancer cells line have performed the ChIP-Seq to see the genome-wide distribution of the two histone modification. And as we can see, a significant difference can be found in the H3K27 trimethylation. And this is a geo analysis. You analyze the triple-negative breast cancer with the control group and find that the different genes was enriched in the cell migration pathway. And this obvious result since the cancer cells have the migration ability, whereas the normal tumor cells don't have that ability. And since the function of the H3K27 trimethylation was silence the gene, and the author particularly interested in the H3K27 trimethylation reduced gene that may involved in the aggressiveness and the metastasis. So they find the CEMIP, which is the gene that can translation to the cell migration inducing protein. And this protein associated with the poor prognosis in cancer. And also it demonstrated indeed a significant decrease of the H3K27 trimethylation marks in the triple-negative breast cancer compared with the normal control group. After they find the target gene, and they used the chemical screen to get this. as we can see. So here is the cell availability and of the tumor cells. And as we can see, the GSKG4 and was the most potent. And the GSKG4 is a demethylase inhibitor. So this chemical-based screen suggests that the H3K27 trimethylation demethylase inhibition supersedes the breast cancer cells growth in vitro. And then they do the in vivo study. And here is the migration assay and the transfer chamber assay. And as we can see, the number of the migration and the invasive cells was significantly reduced in the triple-negative breast cancer cells that treat with the GSKG4. And so those data suggest that the H3K27 trimethylation demethylase inhibition may disrupt the triple-negative breast cancer cell invasion and the migration. And the figure below investigates an anti-tumor effect of the GSKG4 in the human xenograft. So as we can see, the tumor in the group and treated with a higher dose of the GSKG4 has a significantly smaller size in the tumor. And also for the tumor volume, as we can see, the higher dose will have a small tumor volume compared to the control group. And to determine the molecule mechanism of the triple-negative breast cancer cell growth, the author used this heat map and to showing the transcriptional changes in the triple-negative breast cancer treat with the GSKG4 for 24 hours and 48 hours. And we investigate use the RNA-seq. And as we can see, the CEMIP gene was the most downregulated. So the expression level was lower after use the GSKG4 treatment. And here is the geo analysis. And it shows that those differential expression gene was enriched actually in the MAPK pathway. And here to systematically examine the epigenetic changes that are affected by the GSKG4. So they performed another ChIP-Seq of the GSKG4 treatment group compared with the untreatment group. So the ChIP-Seq revealed that the CEMIP promoter has increased the H3K27 trimethylation modifications upon the GSKG4 treatment. And this, sorry, the internet was unstable. And so those are observations and can indicate that the GSKG4 treatment results in inactivation of the CEMIP transcription by increasing the H3K27 trimethylation at the CEMIP promoter region. To assess the expression of the CEMIP in the breast cancer, so their analysis reveals that the CEMIP was highly expressed in the human breast cancers compared with the normal control group. And also, as we can see, a significantly higher levels of the CEMIP was found in the triple-negative breast cancer tumor compared with other tumor. And here is the percentage of the survival rate. And as we can see, an elevated CEMIP level will lead to a lower survival rate compared to the low CEMIP expression. So in the summary, so all those studies can be simplified by using these figures. So in the promoter, in the CEMIP's promoter region, there is a histone modification called the H3K27 trimethylation. So it can silence the gene by condensed the chromatin. So the CEMIP cannot translation to the proteins. And however, if a demethylase occurs, that's the condensed chromatin will be the open chromatin. So the CERMIP will translation to the proteins and may lead to a breast cancer. And the author found that the GSKJ4, which is a demethylase inhibitor, that can prevent the demethylase occur, so that the chromatin will be keep condensed, and the CERMIP will not have a chance to lead the breast cancer. And so here is the overall of the case study. So this brings us to the end of this webinar. So I believe most of you have already know the Novogenes, but some of you may be the first time. So I will give a very brief introduction to the Novogene. And here is our missions, visions, and the corporate value. And Novogene is an international company, and here are our global footprints. And we have cooperated with many leading international companies and universities. And here are our research contributions. So we have powered more than 700 articles that can include in the SAI, and the cumulative impact factor is more than 6,000. And here are the testimonial from our clients. And we have advanced and comprehensive technology platform to meet your large scale and diversified needs. And also the Novogene provides ultra-fast turnaround time from the sample collection to the data releasing, while this fully automated intelligent delivery platforms, the Falcon. So the Falcons is in operation 24/7, and it can significantly reduce the production turnaround time and the labor cost. And also Novogene has the world-class data centers that can provide us the fast, accurate, and secure data delivery. And this brings us to the end of the presentation. So let's move on the Q&A section. So I will pick up several questions and to answer here. And there is a question about whether we accept the cut and run or cut and tag samples. So the answer is yes. So we can accept cut and run samples. And they use the different way to do the cutting, but they will finally give us the DNA fragment. And but we don't have a corresponding pipeline. So it will go through our ChIP-Seq pipeline. And for the cut and tag, we add adapters while using the TN5 transposers. And so we can accept them as a pre-made library. And there is another question about how we charge for the IgG group. So the price is the same as the IP group. And the IgG group usually have a very low amount. So sometimes they cannot be pursued with the live preparation. And we will more recommend use the qPCR to exclude the false positive. And if the amount are enough for our lab preparations, and we can do the sequencing. And we will use the peak calling and the comparison in our analysis. And here's a question about why need RNA removal, since they already used the specific antibody. So in series, only the target RNA can be immunoprecipitated. but there are actually the non-specific binding. And based on our experience, there will be non-specific RNA signal. So we will recommend RNA removal before sending the sample to us. But it's not required, and since the IP samples are precious, so we do not force the RNA removal. As long as you can accept that part of your data belongs to the RNA, and we are okay for this. And here is a question about the bioinformatic analysis. So can we do the comparison for more than 2 groups simultaneously? So the answer is no. So our comparison can only compare between 2 groups, but we will not limit the number. So as long as you think the 2 groups need to be compared, so we can do so. And our webinar is almost an hour, and due to the limited time, let me handle it back to the host.